This is lecture 12 in the course Principles of Metabolism. In this lecture we will look at the metabolism of amino acids and how they become important in nutrient starvation. So amino acid metabolism is a very large topic. There are 20 proteinogenic amino acids and a number of other amino acids that are also important in metabolism, and they have a wide variety of roles and participate in a large number of pathways. So we're definitely not going to cover all of that in this lecture, but we will try to give a bit of an overview. So amino acids, as you know, is an important component of protein, and they have the general structure that you have an amine group joined to a carboxylate group, and then there's a side chain. And the structure of the side chain is really what determines the metabolic properties of the amino acids, its chemical properties. And the variety of side chains is also what gives protein such flexible properties, that we can make so many different kinds of properties out of amino acids because of the variety of side chains. So amino acids generally are metabolized, obtained from diet via protein, or if you grow cells in culture, you generally have free amino acids in the medium, and then you have uptake of amino acids directly from that. They are important intermediates in a lot of cellular metabolic processes. They are substrates for generating nucleotides. It's an important product from amino acids. There's a variety of other amines, things like histamine, for example, which are derivatives of amino acids, and a variety of compounds that contain amine groups come from amino acids. Amino acids are correspond to keto acids in a simple way that you can exchange the amine group with an aminotransferase and put in a keto group instead. And then you get to something that looks more like a carbohydrate. And these feed into central metabolism, the keto acids. And from this you can either form other carbohydrates or you can break them down to CO2 and gain energy. This step between the amino acid and the keto acid is where you have to get rid of the ammonium group. And so in this case you have to form either free ammonia in most cell types, or in the liver this can be converted to urea, which is a less toxic form. So this is the big picture of amino acid, and we're going to look at a couple of particular aspects of their metabolism. Some of the amino acids in central metabolism are essential in humans, and some of them are non-essential. So this reflects whether the amino acids can be synthesized from scratch, so to say, from simpler components in central metabolism. This concept of essential versus non-essential amino acids is a little tricky in some cases. It depends a bit on context. There are several situations where particular cell types require a particular amino acid, whereas the whole body does not, because there are other cell types, notably the liver, usually that can synthesize it so that it exists in blood. There are also some cases where it's a little unclear whether we should classify an amino acid as essential, because there are synthesis enzymes, but in practice they are not very active, or it's hard to acquire enough of the amino acid via the synthesis pathway. So there's a couple of gray sort of areas. Largely, this is the division between non-essential and essential amino acids. If you look at different textbooks, you might find a couple of things that differ. But there are some cases which are gray areas. Methionine is one case which you could sort of ponder whether this is an essential amino acid or not. There is a synthesis step for methionine from another amino acid known as homocysteine, which is not a proteinogenic amino acid, but it is an amino acid. So methionine can be synthesized, but it turns out that this particular process doesn't go very fast, and there's usually not that much homocysteine around in the body, and so this is not quantitatively important, we think. So we quantify methionine as essential. But there are some difficulties to this. But generally this is the classification. So you might wonder why it is the case that about half of the amino acids are essential and half of them are not. So of course in plants and in bacteria there are no essential amino acids. Everything can be synthesized. So there are synthesis pathways for all amino acids in nature. But in humans and in many animals, a lot of them have been lost, but some have been retained. So why are some amino acids essential and others not? So it's an interesting topic, and one pattern you notice if you look through the essential versus non-essential amino acids and what they do is that for the really central amino acids, so the ones which are important for central metabolic pathways that you think really has to be able to function, for those ones the synthesis pathways have been retained. So the non-essential amino acids tend to be the ones which are most important in a way. So this important here, I mean that these amino acids are part of central metabolic pathways. They're an integral part of central metabolism. You can't really remove the synthesis pathways because of that. So here are some examples. Alanine, for example, is an important ammonia carrier, and it plays a role in a lot of aminotransferase reactions. Glutamate is very close to a TCA cycle substrate, also a major aminotransferase donor. Aspartate is part of an important shuttle in central metabolism called the malate aspartate shuttle. We'll look at this a bit more closely in a minute. Arginine is part of the urea cycle. We'll look at that a little bit also. Cysteine is part of glutathione metabolism and something called the gamma-glutamyl cycle, which is also a kind of central process. We're not going to look at that so much. And so on. Serine is part of 1-carbon metabolism, which is the subject of another lecture later on. Proline is one amino acid, which we're not really sure about, but we think it's a redox carrier, and it has some interesting roles in the mitochondria. And there are a couple of more examples. So generally, when you look at the non-essential amino acids, you find them to be part of these type of processes. Whereas the essential amino acids do not. They are more sort of peripheral metabolism and mainly used for protein. So that's an interesting kind of division. So a couple of examples of these central processes where amino acids take an important role. So alanine, for example, is part of something called the glucose-alanine cycle. This is an example of a process which is a whole body process where the amino acid becomes important. So alanine is used a lot as a kind of amine carrier or an ammonium carrier in the body. And this behavior is also retained in cell lines. So you tend to see a lot of cells. This is usually discussed in muscle, but a lot of peripheral tissues and cell types tend to capture excess ammonia by transferring it to pyruvate and making alanine. So alanine is released by a large variety of cell types. And then in the body, the liver has specialized in taking care of the alanine, turning it back to pyruvate. And because the liver is good at capturing ammonia and turning it into urea, which is less toxic, this is a good place to do that. And then the pyruvate can be converted to glucose via gluconeogenesis in the liver. This can go back to peripheral tissues, and then it can be broken down to pyruvate in glycolysis, and then the cycle can continue. There's a couple of variations of this cycle, but this is probably the one you will see most in textbooks. So here's an example where alanine is a central metabolite, not in cellular metabolism in this case, but on the whole body level. So this motivates why the aminotransferase for alanine is important, and its synthesis has been retained. Another example is the malate aspartate shuttle. So this is an example on the cell level. So most cells have this shuttle, and it's a system for transferring reducing equivalents in the form of NADH. So transferring reduced NADH from the cytosol into the mitochondria, where it can be oxidized in the respiratory chain. So as you recall, NADH is, among other things, generated in the cytosol by glycolysis. And if we're running glycolysis and oxidizing the pyruvate we get in the mitochondria, then we have an excess of NADH, and it has to be taken care of somehow. So this shuttle is a kind of intricate system to be able to get the NADH, or the reduced form of NADH, sort of into the mitochondria. NADH itself cannot cross the mitochondrial membrane, but so what the cell does is that it captures the electrons from NADH by reducing oxaloacetate to malate. This can cross into the mitochondria. There it can be oxidized back so that the precious electrons of NADH is recovered. We have oxaloacetate again. But then it turns out that the transport of oxaloacetate back to the cytosol is inefficient for a variety of reasons. So what actually happens is that aspartate is synthesized in the mitochondria via a transaminase from oxaloacetate. And the aspartate can easily travel back into the cytosol, and the same transaminase actually, but in the cytosol, converts this back to oxaloacetate, and now the cycle is complete and we can continue. It's a kind of complicated system, but an efficient system for the cell to transfer NADH into the mitochondria. And because of this, the synthesis of aspartate is now an integral part of central metabolism, and so its synthesis has been retained. Now, we'll look a little bit at what happens to amino acids when there is a lack of nutrients, a nutrient starvation condition in cells. So, because amino acids constitute protein, and protein is a large component of the cell biomass, this is a possible source of energy in cases where we have a sort of nutrient crisis and there's no other way for the cell to get the energy it needs. So, as we said, catabolizing amino acids can yield keto acids, and we can go on to make carbohydrates, and we can catabolize this down to carbon dioxide, and we can get energy from it. It's not a perfect substrate because we have this problem with the ammonia that we have to get rid of, and ammonia itself is toxic. So, it's not the perfect substrate, but if we are out of sugar and we're out of fat and there are really no other options, then this is a kind of backup system. So catabolism of amino acids is heavily regulated by nutrient sensors, energy sensors, that kind of thing. And when the cell is sensing that there is a lack of energy and sort of a crisis, then what happens is that all the anabolic pathways from amino acids turn off. So protein synthesis is shut down largely, and instead the amino acids are liberated from proteins. Biosynthesis of things from amino acids are downregulated, and the amino acids themselves are preferentially converted to keto acids and catabolized, and the synthesis of amino acids is turned down. That's a reasonable response. That's what you expect would happen, and that's indeed what does happen. And so when amino acids are converted to keto acids, there's a few different fates. In many cell types, they are just oxidized so that we can get energy from it. In some specialized tissues, like in the liver, they are used for gluconeogenesis so that the whole body can survive, so that we can have sugar in the bloodstream. They can also be used for ketone bodies, which can be used instead of glucose in some tissues. So this is the overall picture of what happens to amino acid metabolism during nutrient starvation. So all the amino acids, the proteinogenic amino acids, all of them can be catabolized completely to carbon dioxide and ammonia. And generally, you can obtain not as much energy as you get out of glucose, but maybe 70% or something like that, weight per weight. So you can get a fair amount of ATP from amino acids. There's a whole set of catabolism pathways, and there's lots of enzymes involved, and it's very complicated. What is shown here is an overview of the proteinogenic amino acids and how they are organized. And so, as you can quickly see, is that all of these amino acids, they can be converted into each other in some cases, and then at the end, these can all reach the TCA cycle by one route or another. So there are metabolic pathways that allow any of these amino acids to convert their carbon and send it into the TCA cycle. So for example, asparagine can be processed to aspartate, and that can yield oxaloacetate, and then you're in the TCA cycle. Or methionine can be converted to cysteine, and cysteine can yield pyruvate, and now you're in the end of glycolysis, and you can get acetyl-CoA, and now you're in the TCA cycle, and so on. So there are routes for all of these, and if you're interested in particular amino acids, it's well, you can look these up. But that's important to know, that all the amino acids are possible to catabolize. So we can really make use of all the energy that is stored in protein in terms of the carbon skeletons of these amino acids. So it can be a bit bewildering to look at all these pathways. So one helpful kind of pattern is that it often helps to locate an amino acid by looking at its corresponding keto acid. So in some cases there are easy, obvious ways in which an amino acid can feed into central metabolism, because if you just look at its corresponding keto acid, so having the amine group exchanged for the for a keto group, then you immediately end up with something that you might recognize. Alanine, for example, is just one transamination away from pyruvate. Glutamate immediately yields alpha-ketoglutarate, which is TCA cycle metabolite. Aspartate yields oxaloacetate. These are some of the most central examples, but in many cases you can sort of locate the amino acid in metabolism by looking at this. So amino acids are not that strange. They are just one amine group away from a carbohydrate, really. Now, in order to get at the actual amino acids, cells have to break down protein. And this is done in a variety of ways, but an important process is the one called autophagy. So this is literally self-eating. So many cells have a machinery whereby they can target certain parts of their own structures, their own content, for degradation. So this is a large and complicated field, and we're not going to talk about this much. But it's good to know that there are these systems where large structures of the cells can fuse with this type of kind of lysosomal-like vesicles. And in these you can degrade proteins and also other material, and this can liberate amino acids. And this is of course regulated in a variety of ways, so that structures are only broken down when needed. And to the right here you see an example of this in a mouse hepatocyte. So in this case, these cells start to degrade their material after a day of nutrient starvation, which is a long time for a mouse to be deprived of nutrients. So these cells are really energy poor, and they have now switched to accessing protein and their own structures for catabolism. See, this is what it looks like. In some cases, the amino acids are obtained from endogenous proteins. In vivo, in some cases, there are other tissues that degrade protein and sort of send it out for use by other cell types. And the classic example is that muscle, which is very protein rich, degrades a lot of its protein when required and sends this out. So this is a kind of interesting system in itself, because when people looked at the actual amino acids that are supplied by muscle by degrading protein, it doesn't look like muscle actually just hydrolyzes its protein and sends out all the amino acids. There is a very controlled conversion of amino acids into a few central amino acids, which are then sent out into the bloodstream. So alanine and glutamine are thought to be major amino acids, which are produced by muscle in cases of nutrient starvation, and which are taken up by other tissues in the same conditions. So there is, in this situation, there is a highly regulated response where protein is broken down and amino acids probably feed into central metabolism, and a couple of selected amino acids which are good as nutrient carriers and energy carriers are secreted in response. So that was a very brief overview of amino acid catabolism, protein catabolism in nutrient starvation. Finally, we're just going to look a bit at what happens to all the ammonia that comes out of these processes. So the central sort of ammonia waste disposal system in the body is the urea cycle. This is a system that is confined to the liver. There are enzymes in this cycle. There's 4 central enzymes, are found in various other cell types and tissues, but you never find the whole complement at the same time and functional except in the liver. So the purpose of this system is to capture ammonia, which is a toxic metabolite at high concentrations, and convert it into urea. So urea is a form of ammonia bound to one carbon, sort of carboxylate group. And this form of ammonia is very non-toxic. You can reach very high concentrations of this without affecting anything. So it's really an inert metabolite. So this is a very good form of ammonia to be carried and excreted. So how does this work? Well, so we can see there is a cycle. There's a few amine-carrying amino acids involved in this. There are 2 routes of ammonium entry. One is called the transamination route, and so here ammonia comes in the form of transamination reactions, and it's captured as aspartate. And in this case, the aspartate is channeled into the cycle by forming first an intermediate called argininosuccinate, where the aspartate part is here, but the actual amine group has been transferred. And then this part of aspartate, which is actually like fumarate, is then cleaved off. And in this way, just the amine group is able to enter. So that's one way of getting ammonium into this system. The second route is sometimes called the deamination route. So if you have free ammonia in the cell, which you get from some reactions, there is a mitochondrial enzyme that can capture this and sort of fuse it with carbon dioxide, sort of carboxylate the ammonia. And it adds on a phosphate as well, and you get a metabolite called carbamyl phosphate. So a small metabolite, just this amine group, the carboxylate group, and then phosphorylated. And this metabolite can be fused to an amino acid called ornithine. And so when that happens, it gets sort of tagged onto this side of the amino acid, and you get another amino acid called citrulline. So that's the other way you can get ammonium into the cycle. And then the cycle is completed by arginine, which is formed through this process, undergoes a kind of cleavage reaction where the actual group towards the end here, this part is cleaved off and you form urea. And then you can restart the cycle again. So we see that there's actually 2 sites of ammonium capture. There's one here and there's one here, and that corresponds to the 2 nitrogens that are cleaved off in each sort of turn of this cycle as urea. This cycle is energetically costly. A couple of these reactions require ATP. I haven't put in all the details here, but in the end you have a cost of about 3 ATP to produce 1 urea. So about 1.5 ATP to capture an ammonium group. And so this is part of the reason why it's not as efficient to burn amino acids to get energy as it is to burn glucose or fat, because there's an energetic cost to handling the toxic ammonia. And so that makes it not as great a substrate. But with this system, the body is still able to burn amino acids and handle the ammonium in a safe way. If you look at amino acid catabolism in cell systems, then you don't have a functional urea cycle, and cells will just release ammonium. In some cases, they will attach it to alanine or something like that and release that instead, but you will not have urea production.